I am so excited that everybody's here. So New Orleans is our hometown with AxoSim, so we're really happy that everybody's here. I hope everyone has a great time, but also gets to see some fantastic scientific talks and see where our field has come really a long way in the last few years and where we're heading now. So my talk is going to concentrate on our NerveSim platform at AxoSim. So Stefan mentioned the CNS models, but I'm going to sit here and talk about the peripheral nervous system. So briefly, again, we're located here in New Orleans. We have a bunch of our team members here, which is fun. So we were started as originally spun out of Tulane University as a platform developer, both in neurodiscovery and neurotoxicology. We've also now expanded into some CNS applications with the platform we licensed out of Johns Hopkins, out of Thomas Hartung's lab, and we have a growing team here of 27 people that are working on these platforms. So again, I'm going to focus mostly on NerveSim here, which is our PNS platform, but I mentioned the BrainSim as well. So the PNS is really we see as a bit of an under-researched area, but really in the past couple of years we've seen a big resurgence, which we've been really excited about, both on, there's a big issue with off-target effects with a lot of different drugs, especially in chemotherapeutics. Also happens with diabetics, with peripheral neuropathy. But we also now are seeing a big increase in terms of drug efficacy work here. And a big reason for that has been the work that Disarm Therapeutics, which is now Eli Lilly, was doing here and discovered a new pathway that really regulates a lot of peripheral neuropathies and axonal degeneration in general. So we always think of the nervous system as our brain, of course, and the CNS is really the brain and the spinal cord. Peripheral nerves are everything outside of that. So the peripheral nerves are how the brain communicates both with our body but also with the outside world. So it's extremely important, and there are so many different applications outside of even just the tox side, but also in a lot of different disease indications. So chemotherapy and diabetic we may not think of as an actual disease indication. However, there are now a lot of therapeutics that are being developed to be able to either protect from that or regenerate following the incidence of these. There are a lot of genetic peripheral neuropathies that are now being looked at more in depth that we're excited to be working more in. And then also ALS, which is more on the motor side of the PNS as well. So I'll talk here now about NerveSim. Where it really starts for us is being able to actually mimic the neuroanatomy of that peripheral nerve. So can we actually mimic what we saw there in that previous diagram of these, where all the cell bodies are at that dorsal root ganglion by the spinal cord and then extend axons outward to the periphery. Now, we do the same thing with this model. We have all those neuronal cell bodies in this bulbous area on the left, and you can project those axons down up to 1 centimeter long. There are two main things that are really, what I think really differentiate this model and what we've focused on in making this a clinically relevant model. That is, one, the fact that we get Schwann cell myelination. So we have the glia in the model itself, and they do migrate out from that end and can align with and then wrap the axons and actually deposit that myelin down around those axons. And also the fact that we can look at population level electrophysiology and run nerve conduction studies. So nerve conduction studies are what is done in vivo, also what is done in the clinic, and we can do that now on an in vitro platform. Briefly, what that looks like. So we can take different cell populations. This could, I mentioned, of course, neurons or sensory neurons with Schwann cells. We can do it motor neurons and astrocytes. It depends really on the application. We then take those co-cultures together, put them into a spheroid. Now, there's a lot of spheroid work that's done in a lot of different areas, but then we take that and put it onto the NerveSim construct. Now, over the course of, again, depends on the cell type, but it could be up to 4 weeks, sometimes even longer, sometimes shorter. We then can grow and really guide how those axons grow down this channel, and then now over electrodes as well, to be able to then go in and do this population level electrophysiology. Again, these readouts look very much like what you would see from an in vivo readout. and we're talking now on the order of tens of thousands of nerves, of neurons and axons in here, which is, again, what allows us to look at these field potential recordings. And that same architecture that allows us to look at that level of detail in terms of electrophysiology also gives us the ability to look at histopathology, and even at the electron microscopy level. Here you see that nice compact myelin. I mentioned some of these readouts, and I'm going to briefly touch on a few of these points here, but we have another talk tomorrow that Lowry Curley, our CEO, will be giving on a specific case study, and then Corey Roundtree, our lead scientist, will be talking about a project he led on really scaling this platform up, which is quite relevant to the discussion that we were just having next door, too. But there are a number of different endpoints we can do to be able to multiplex these as well, which is extremely important, of course.
Could you say that again?
Siri's picking up on my talk, too. This is some of the foundational work that looked at really being able to characterize the myelin on the model. And we could see the myelin basic protein being present on quite a few of the axons in the preparation. We could see that mature over time. We also see that manifest in terms of the electrophysiology, both in terms of stronger signals for both amplitude and conduction velocities when comparing the myelinated preps and the unmyelinated ones, which we can induce with our different growth techniques. Again, won't go too deep into this because Lowry will be presenting more of this tomorrow in a fuller case study, but we've looked at this in terms of from a toxicology and a peripheral neuropathy off-target effect standpoint, seeing dose responses with some of the key chemotherapeutics out there that are known to cause issues, and we've seen, and we've run it with controls. We've even been able to look at, I think it's actually originally Stefan, when doing work together years ago, said, well, these dose responses are fantastic, but how do we know that just throwing a high enough concentration of a drug on these isn't just killing all the neurons? So we then went back and were able to develop a viability assay on top of this, and now what's really nice is we can be able to delineate between axonopathy versus neuronopathy and see, are we seeing axonal degeneration or are we actually seeing cytotoxicity at the neurons themselves? Then once we know if the kind of binary, are they toxic or not, we can start to dive deeper into mechanisms. Is this, as I mentioned, the neurons that are being affected? Is it the axons? Is it demyelinating, or is it more general axonal degeneration? This is also really important in terms of some of the disease modeling implications. For instance, like a Charcot-Marie tooth, which is a genetic peripheral neuropathy where there are two general phenotypes. One is more axonal and one is demyelinating. This is something that we're looking more deeply into now. And I also mention this because in some of that data we just showed, those were all small molecules. And we're talking, and again, talking about the discussion next door earlier, there are a lot of, and where I think a lot of the fit for purpose uses of these systems are going to be, are other modalities. So we've done small molecules, we've done larger biologics. In this case, this was some early work we did with some AAV9 work because we wanted to see, is this a suitable system for looking at gene therapies? And in this case, this is just with a GFP as the reporter, and it was phenomenal for us to see and really exciting for our partner we were working with on this to see that basically we're seeing really good transduction of that GFP into all the cell types there, not just at the neuronal cell bodies, but all the way down the axons and even in the Schwann cells down the channel. So we've actually done this in a few different species, but then also a variety of different iPS cell types. And we've now developed a really good system in terms of how we characterize a model with new cell types. So a lot of time, if it's an iPS cell type, we'll start in 2D, really develop the right media, the right soup to grow each of these different ones, and then we'll move into 3D, and then ultimately onto the NeuroSim platform. Primary tissue is a little bit of a different process, but still has its own process in terms of how we get that tissue and then form spheroids and put them into the system. So I mentioned we start with 2D, but then we'll move into kind of a 3D spheroid, as you saw in that animation, before we move it into the NeuroSim platform. And what we've started to see these amazing images. These are pretty hot off the presses, so I'm excited about a lot of this work. And we have So these are some commercially available sensory iPS-derived neurons with our own internally derived proprietary Schwann cell, human primary Schwann cells. We're seeing amazing alignment here. We're seeing bundling of the axons here, and now we're putting that onto the NerveSim platform. And again, this is really new data, and this was really exciting for us because what we're seeing on the right is, well, in the green we're seeing S100 for Schwann cells. On the right is myelin basic protein to actually see the myelin, that these Schwann cells are functionally myelinating. And this was after only 1 week. So I mentioned typically we grow these out to 4 weeks. So hopefully in Berlin next year we'll have a full case study on the new cell types here we'll be able to show, but we're really excited about where this is heading. In terms of being able to use this in a disease modeling setting, we have been working on a grant, and Lisa, who's here, is leading the work on with a congressionally directed medical research program on using the system for ALS, and specifically the SOD1 variant of ALS. And we saw some really nice early indications that we're seeing some protein inclusions in there, and now we're kind of diving deeper. Are these truly Lewy bodies? Or is this, it looks like the pathology of ALS. We've also published on what was at the time the first human Schwann cell on human iPS neuron myelination, and that has. Do I need to end my time? Anyway, so just kind of further lending itself to being kind of a scalable platform in terms of different biology that can go into it. We'll also talk a little bit, we've been doing some work in pain, and our academic collaborators, as well as Wesley, one of our scientists who's here, worked a lot on being able to use this in a pain setting, but also as a synaptic model. So can you have the dorsal root ganglion now synapse onto the dorsal horn to have that primary synapse, which is where those mu-opioid receptors are located as well. And I'll quickly go through some of this just because this is really fun research, and this was a phenomenal phenomenon that we saw was, so that we know the dorsal root ganglia alone, when they extend their axons down, then they showed with the dorsal horn alone extended axons down in the opposite direction. But when you co-cultured them, we only saw unidirectional growth of the DRG toward the dorsal horn, which is really important because that's what happens in the body. And so that then allowed us to look at, from an electrophysiology standpoint, stimulating at the dorsal root ganglion and recording postsynaptically at the dorsal horn to be able to look at that pain circuit. And now we're just getting started with what where we'll be able to go with that. And now that work was actually started with rat. Now we're moving into, done it with some human work. And now where we're going with all of this is, and again, kind of coming out of that conversation next door was really the fit for purpose and throughput. A lot of what we were doing was really lower throughput, and it was really good in being able to use it in a mechanistic and investigative toxicology type of a setting. Now we need to get that throughput up so we can get into discovery applications for it, and that's essentially what we're now doing. So we developed internally we call it an embedded electrode array system that is specifically designed for peripheral nerve biology, and again works for lower motor nerves as well. We have 10 electrodes along this chip. Again, I'm not going to get into too many details because Corey's going to present a lot of this tomorrow. But this is done over a flexible printed circuit board and now on a 24-well chip, so that we'll be able to run these in parallel, a lot of different tests. We can do longitudinal studies, and it really allows us to not just treat with a neurotoxicant and see if there is a deficit, but now can we induce that deficit? Can we co-administer something in terms of being able to look at it from a neuroprotection study standpoint, or can we then actually look at nerve rescue as well from a neuroregeneration standpoint? And what's been phenomenal with this is the level of data that we can pull out of this. So we can look at we're now seeing off this platform multiple different fiber types and multiple different conduction velocities in the same prep. So where this gets really powerful is, now if we can start to see a deficit in one particular area, is that tied to a specific ion channel? Is that tied to a pathology that is manifesting in that from a disease line? And we're seeing also, because of the platform, we can do current ramping, much like you do in an in vivo study. And so now, can we stimulate certain neurons based upon the electrophysiological parameters that are being input there? We can block those and then rescue them again and have them come back. And what was interesting in this study was this now sets the stage for us doing even a sensitization of the neurons to be able to do a number of different studies with it. So in summary, I know I didn't go way in deep on any of this, but hopefully Lowry's case study tomorrow and Corey's poster didn't want to steal too much of their thunder. But I'm really excited about what we've done here. We've validated this from an induced peripheral neuropathy standpoint, now really moving into using it in a drug discovery setting, again, more on the efficacy side, both with CIPN and diabetic peripheral neuropathy, but then also with patient-derived lines in different areas, pain and ALS. And now we're actively going into partnerships with these models with clients and partners. So again, I'm so happy that everybody is here in New Orleans. Please come and see us and talk to us, not just about what we're doing, although this is mostly what we want to talk about, but also if you want any tips on what to do while you're in New Orleans. It's a great place to have a meeting, but it's also a great place to do some really cutting-edge research. And I would be remiss if I didn't say that we are hiring, and if you want to live in this place and do really cool research, we're here. So anyways, thank you very much.